Okay, welcome back. I hope you had some meaningful discussions and that have managed to clarify some of the issues which were not as clear to you during the presentation. So we now want to move on to the third part, the third and final part um, of my talk for today on health-based exposure limits in clinic validation, an overview and introduction. So the first part, we looked at the context. In the second part, I tried to introduce the issue of health-based exposure limits. And in this particular part, which is part 3, I wanted to talk about some practical things to consider. If your organization has decided that this is the way that they want to go, perhaps you're thinking of, contracting someone to do the work for you, etc. And then I just want to say some things which come from a more practical position to help to tie up some of the things that we're talking about. Okay, so the first thing which I want to say right from the outset is that the calculation of health-based exposure limits is not a matter of guesswork. I have there a cartoon where we've got a pharmacist speaking and saying he's still alive. How you guess his doctor's handwriting, I'll never know. But in essence, what I take as the take-home message from that cartoon is that, you see, pharmacists are trained to interpret prescriptions. So what might seem like, is just looking at handwriting and dishing out drugs, there's got some other processes behind it. And it's the same thing with the calculation of health-based exposure limits. It's not just a matter of saying, oh, okay, intraspecies, variability, I'm going to put a 10. Oh, interspecies variability, I'm going to put another 10. Oh, duration of study, I'll put another 10, and things like that. Because it has to be a structured scientific evaluation which takes into account all available toxicological and pharmacological data, both preclinical data as well as clinical data. And you'll find that as you spend more time doing these calculations, you start to have an appreciation of why this statement is important. And actually, that's probably one of the reasons why in the guidances, they always talk about a qualified expert, which is something what I'll also talk about, I think, in the next slide. Because it's not guesswork, it's not just plugging in figures. There is a scientific basis to it. There's some professional judgment that comes to it. So the data and information should be gathered and critically evaluated by a qualified expert. So it's a good idea, if you intend to have these monographs done for you, that you actually get a qualified expert. You see what the problem is? The problem is, even in doing these calculations, it could cost your organization lots of money if an overly conservative approach is taken where numbers or figures are just plugged in. Because, mind you, the adjustment factors that we saw earlier, especially from the guidance from EMA, can go up to 10, and you've got 5 of them. So it means you can get up to 10 to the power 5. And what is that? That's what? Is that 10 times 10, 100 times 10, 1,000 times 10, 10,000 times 10, 100,000. So you could actually get very small values in terms of the limits which are set, which would then make it difficult for you to have analytical methods which are sensitive enough to pick up some of the values that have been calculated. And whilst the regulator might be fine with it because it's an ultra-conservative approach, it might actually cost the organization lots of money. So who is a qualified expert? The definition of a qualified expert, this one is, I think I took this one from the EMA guidance. It talks about an individual who's got a background, an educational background in toxicology, pharmacology, or related health sciences. And this is extremely important if someone is going to evaluate the information. because you want somebody who's got an understanding of when some data may not make sense from a human perspective, and someone who's got an intimate understanding of the various type of studies, experimental studies, which are used in the field of toxicity, of toxicology. Like a simple example would be if you're going to be evaluating data from a DAT study, looking specifically at embryo toxicity or teratogenicity, that if the study has not been conducted in such a way that the compound of interest is administered during the time of organogenesis, then you can actually miss and have very wrong results. And certification is a good idea. Somebody who's got some internationally recognized certification, like the Diplomate of the American Board of Toxicologists or European Registered Toxicologist. And basically what this does is it gives you peace of mind, if I can use that in quotes and loosely, that the person who is a toxicologist is actually a real toxicologist who has been evaluated and viewed by his or her colleagues as someone who is competent. And also there must be the issue of adequate experience in the practice of deriving health-based exposure limits. So unfortunately, even though risk assessment is a part of toxicology, there are some programs where risk assessment is actually not taught. So one of the things that you might want to do from your organization is to make sure that the toxicologist that you are hiring has either taken up courses in risk assessment as part of their postgraduate training, or if it is on the job training, has attended courses specifically looking at risk assessment. There are a number of summer schools which are run ordinarily for 40 hours or more, which is close to a week of intensive learning and training, where students are introduced to the issue of risk assessment and can actually practice most of these techniques that we use in doing the risk assessment. So that's really a good idea. So this is adequate experience in the practice of deriving health-based exposure limits, such as occupational exposure levels, PDEs for residual solvents, elemental impurities and product contaminations, etc. And that's very important. And one thing that you have to define or to make a choice is whether you will need to have an internal toxicologist to work on all your APIs, all your new compounds, or whether you're going to get a contract toxicologist to outsource it. So, assuming that you do not have an internal toxicologist, as most organizations would not have internal toxicologists, what do? Well, you can actually outsource or acquire PDE monographs externally. But even if they are acquired externally, best practice and current guidance entails that you ensure that they are reviewed internally. So ideally, it's a good idea that the PDEs are reviewed by someone who has got some level of expertise in terms of a health-based exposure limit derivation, so that you can be able to look for the red flags and see what, whether the monograph you've got is a very good monograph or it's just a useless one, a waste of your money. The other thing that you want to do is to ensure that, when you get a monograph, it actually applies to your context. If you are a pharmaceutical company which does not manufacture any, IV compounds, you do not have any parenteral compounds, perhaps you just manufacture oral compounds, or compounds for oral administration, and then, some of the PDE calculations, which would talk about the intravenous route may not necessarily be pertinent or apply in your particular setting. So a good monograph should be one that applies in your own setting. And then one thing that you also want to do is to make sure that when you purchase these monographs, the suppliers should commit to answering regulatory queries should any arise during any inspections. It should not be, like you've just bought bread and gone home and eaten it, but it's a relationship, it's a contractual relationship that when the regulators come and there are some issues which are pending, they should be able to get in touch with the external contractor that you worked with to clarify whatever issues require clarification. In my perspective, it's a good idea to negotiate for your staff to be trained in terms of these health-based exposure limits. The fact that you're listening to this already in itself is a very positive step, because at least now you have a general idea about these health-based exposure limits. But it'd be good to have staff trained on the basics, and have a qualified expert to review and sign for the PDEs that you may actually develop in-house if your staff have been adequately trained. And you could talk, to some to some expert toxicologists or people with the expertise in the derivation of these monographs to see how to go about this. And then if you've got any idea, certain direction that you want to take, it's always a good idea for you to discuss whatsoever steps you want to take, your planned actions with the regulator. A third thing that I want to talk about, I just want to give you some tips, some things just to be careful of, some red flags to look out for. Be careful of cheap PDE determination strategy documents or monographs, because out there, I am aware that there are some which are ridiculously cheap. The amount of time that it takes to derive these monographs, it's quite significant. And oftentimes, a monograph which has been done well is authored by a person who's taken the time also to purchase some articles or papers to make sure that the evidence that they are presenting in the monograph is actually correct based on what is published out there. So be careful. Cheap monographs, small ones, 1 or 2 pages, they might prove to be more costly in the long run, as the regulator might actually see right through this. One thing that you should also look out for, if you get a monograph and they are using LD50 for the derivation of PDE, that's a red flag. I think I've talked about this already. That when you're doing the calculations for PDE, you do not want to use acute toxicity data because of the very definition of PDE, or the very definition of a health-based exposure limits is talking about a lifetime. It would be really hard to even bring in some adjustment factors, because the way that an organ, or an organism responds after an acute exposure to very high doses may be totally different to the way that it would respond after chronic exposure. At much lower doses. And then another red flag that you should look out for is if you see your PDE values are calculated using values from material safety data sheets as a main source of data. That's also something that you worry about. Because ideally, when you come up with these monographs, you want to use primary literature, primary data, like the actual studies which we used. At the least, maybe abstracts, but it may be data from reputable, organizations like ECHA, FDA. But MSDS is, then you should also be a bit worried. And then another issue that can raise or should raise a red flag is if in all the PDEs that you get, the adjustment factors are all the same. That every time adjustment factor F1 is 10, F2 is 10, F3 is always 3, F4 is always 1, F5 is always 2 or 6. That's something that should also start ringing bells in your head, because this is a case-to-case analysis where each and every compound is viewed individually. And then also be worried, or not really worried, because sometimes it is possible that you can use a single approach, but generally you expect there to be at least 2 approaches. If there's sufficient data in animals, we expect at least one approach which is looking at animal data, and then then another approach may be looking at the clinical data, ordinarily the point of departure being the minimum or lowest therapeutic dose. And then also generally you should be worried when you're dealing with human or human medicines or pharmaceutical substances, if all the approaches make use of animal data. That might be a big issue. And then also if there's over-dependence of what I've termed weak data sources, like it seems like the whole monograph is based on abstracts or is based on anecdotal information. Those are things that you generally want to look out for. Fourth thing, so PDE values will actually differ generally. So risk assessment for those who have done toxicology has been defined as both a science and an art. Because if you remember in one of the formula, we had what was called the modifying factors, which then bring into perspective the issue of professional judgment. And even in the derivation of, in the use of the different adjustment factors, F1 to F5, there's always the issue of, your professional judgment. And now, depending on the type of studies which are used by different toxicologists and what they've defined as key studies, you can find that the PDE values could actually vary up to about 10 times. Although, of course, current guidance talks about 3, but it is not abnormal to actually get 2 PDE values which are varied up to 10 times. One is 1 milligram per kg body weight per kg, 1 milligram per kg, 1 milligram per day, and then the other one says 10 milligrams per day, which is actually perfectly normal. You get worried when the variances now go up to 30, 20, 15, going on like that. And the reason for these variances, like I said, it depends on what is used as the key study. One person may use as a key study perhaps a study looking at embryotoxicity, and somebody else uses a study looking at carcinogenicity. And if the NOAEL values of those are significantly different, then you could also get different PTE values coming up. And again, I've mentioned the issue of clinical data. The PTE values could also vary depending on how the clinical data was used versus animal data. And generally, we expect that for drugs used in human beings, there's a more dependence on clinical data than there is on animal data. And actually, you do find that when you do many calculations with clinical data, the PDE value is normally lower than that which is used in animals. Well, obviously that makes sense because when we do toxicity studies in animals, we normally work with doses which are much, much higher than therapeutic doses, therapeutic dose equivalents that is used in human beings. And talking about the art, there's the issue of how conservative the expert is. There are some toxicologists who are a bit more conservative compared to others. Not just how conservative the expert is, but also how conservative the organization or the company that wants the PDE done is. There are some who are extremely conservative, whereby the adjustment factors, higher values are used for them, and then there are some who are a bit more conservative, where lower values are actually used. So, like I said this before, that according to PICS guidance, they say a threefold differences are normal, in that 3 to 10 may be justified, but generally anything above 10 is probably not justifiable. Yeah. And then on to some final thoughts as I come to the end of this part 3. So some of the adjustment factors are interrelated, and this brings me back to the first thought which I put out, the fact that you do not just plug in values. So because they're interrelated, careful thought must be used when putting in those adjustment factors. Because if you don't do that, you can have what I've defined there as over adjustment, where you've got adjustment factors up to magnitudes of 5,000, 10,000. Generally, when you go above 3,000, you start getting worried that maybe the data which is available is not as accurate, efficient, is not as accurate and sufficient as it should be. So it's a good idea to think through that. And you can avoid this issue of over-adjustment by having someone who's experienced in the calculations and also having someone who is actually qualified to do these calculations. The more uncertain you are, the larger the uncertainty factors, and therefore the smaller the PDE value. The smaller the PDE value, it means you need to have more sensitive methods developed. in your organization or in the lab, and those are actually a bit more expensive in the long run. And then the other issue, I mentioned a point of departure. And point of departure, generally, for what we've been seeing from the different calculations, it's normally the NOIL value which is used as a point of departure. So point of departure is probably a statement which is used for the value that will then be used in the calculation for the PDE or whatever health-based exposure limit. And like I said earlier, when you're dealing with therapeutic drugs, ordinarily therapeutic dose should be used as the point of departure, because this drug is already being used in human beings, and therefore, the data that we're getting is perhaps more relevant. And just to conclude again, I want to reiterate this point that it's a good idea always to use a qualified expert. So having said that, I think we've come to the end of this talk, and I just want us to just go through those objectives and see if we've managed to meet some of them. So by now, I hope everybody has had a revision, and you can actually be able to state the sources of cross-contamination during the manufacture of drug products. We saw that in the first part of this talk. We also talked about the 3 historically based methods, the 10 parts per million, the 1 over 1,000th of therapeutic dose, as well as the LD50 divided by some adjustment factor, and what the limitations of these methods were. And we've mentioned PDE, we've mentioned ADE, we've mentioned POD, we've mentioned HBEL, we've mentioned NOIL, we've mentioned NOL, we've mentioned LOIL, and LOL. So by now you should be able to know what these abbreviations are. We spent a bit of time just talking about the different methods for calculating health-based exposure limits, which I hope also you have an idea about them now, and then you have an idea of how these health-based exposure limits are actually calculated. And then in the third part of this talk, we discussed some important issues to consider from a practical perspective. The issue that it's important to have a qualified expert do these things for you. That PDE values could differ. If you're purchasing monographs, what are some of the things to look out for? So yeah, I hope this lesson was useful to you. And I spend just go back to groups. I spend some time just discussing the things that we were talking about now. If there are any questions, I'll be happy to answer the questions, and we'll see how best to move. And I look forward to meeting some of you in the more advanced courses, where we actually look at how to do the actual calculations of these PDEs. But as for now, thank you so much for your time. Get into your groups. Let's talk. Let's discuss. I hope you learned something from today. Professor Tagwireyi, I was happy to have you with me. Until meet next time. Au revoir.